I think last time we ended up here with propionic acid fermentation. Okay, what you have to remember about this, propionic acid, Propionibacterium, as an example of a bacterium that performs this type of fermentation. Now, the last one we need to think about is mixed acid fermentation, which is used by really quite a lot of bacteria, mainly many enterobacteria. So Escherichia coli performs mixed acid fermentation. It's a big here. Come back. Oh. So what is going on here? Okay, let's have a look at this. If you have a look at this slide, what we've got, we start out with pyruvate, and the first step is going to be cleavage of pyruvate into acetyl coenzyme A and formic acid. So this is by an enzyme called pyruvate formate lyase that Escherichia coli has. Now, if you remember, or if from your Krebs cycle, normally to convert pyruvate to acetyl coenzyme A, the enzyme is pyruvate decarboxylase. Yeah, I believe. But that will put 2 electrons onto NADH, which are then used in the electron transport chain. But the whole point of fermentation is to recycle NADH to NAD+, if you remember. So it would be counterproductive for E. coli or other enterobacteria to use pyruvate decarboxylase to convert pyruvate to coenzyme A. So pyruvate decarboxylase, pyruvate plus gives you acetyl coenzyme A and 1 molecule of CO2. So that's why the first enzyme in the pathway is this pyruvate formate lyase. And then from acetyl coenzyme A, then you get a bunch of different fermentation pathways or metabolic pathways. And the end result is a bunch of different products, acetate, ethanol, isopropanol, butanol. And at different stages of the process, you can use up NADH and regenerate NAD+. That's the basic function of fermentation. And the added bonus of this is that some of these steps allow bacteria to generate some extra molecules of ATP. So low ATP production is the big drawback of glycolysis. So mixed acid fermentations are more complicated than the previous ones we saw. Every time you see something complicated in biology, you have to think, why is it so complicated? Things aren't, well, things are always complicated for a reason. Okay, and here the reason is you can get some extra ATP out of the process. So it makes it worthwhile for bacteria to, evolve all these complicated metabolic steps to actually be able to get a couple more of ATP molecules. Okay, so mixed acid fermentation. Okay, so you have several acidic byproducts like acetate, formic acid, butyrate, and also some alcohols like isopropanol, butanol, ethanol. So if you look at the production of these different products, you get a bunch of them produced at the same time. Now that has got some rather interesting consequences. One of them being that, we all have these bacteria in our guts and they are all fermenting carbohydrate that we eat, starch and sugar that we eat. And so some of them are going to be producing ethanol. So that means, depending on your diet, you probably have a small concentration of ethanol circulating in your bloodstream. You can't have absolutely zero because it's produced naturally by gut bacteria. And this can actually be a problem for some people. And it was like this was brought to light with a case in America. There was a woman driving her car, and the police stopped her. They drew her over to the side because she was like driving erratically. They're like, "Step out of the car, please, ma'am. Blow into this bag," and she was positive for a blood alcohol test. And she was outraged. She said, "No, I've been drinking at all. I don't drink." And it was true. She was telling the truth, but she'd had some kind of meal with a load of sugar, and the bacteria in her gut like fermented a lot and produced a lot of ethanol. So that's kind of be aware that this is a real thing, and depending on your gut microbiome, you can actually have a non-negligible concentration of ethanol in your bloodstream. Okay, now another thing that is pertinent here is that the first step, okay, the pyruvate to acetyl coenzyme A produces formic acid. So this is the main acidic byproduct of these mixed acid fermentation pathways. And the drawback is that the pH is going to go down very quickly, as we saw in the lab class, just yesterday for some of you. And Escherichia coli is not is a mesophilic bacterium, neutrophilic bacterium, so it doesn't grow well at acid pH. So as the pH goes down, what E. coli will do is produce another enzyme, which is this one, which is called formic acid lyase, I believe. And it's going to split formic acid into molecular hydrogen and CO2. So at alkaline pH, okay, formic acid is going to be one of the main fermentation products, but as the pH goes down, E. coli will produce its enzyme and split up the formic acid into molecular hydrogen and CO2. And then at lower pH, then you get an increase in the production of carbon dioxide, much higher than here, and hydrogen gas. Okay, so a lot of gut bacteria will be producing dihydrogen gas. Okay, so let's have a little bit of a reminder about fermentation pathways. This is pretty much the end of this. So I'll just open this question for you. Let me stop. Reinitialize. Okay, so now it should be open. Okay, I've got a lot of votes in. And let's see what you got. Okay, so here are the votes. Okay, number 4 is correct. That's right. This is a major drawback. End products can be toxic. Acids, ethanol, toxic byproducts. These can limit bacterial growth. So it's a problem if they build up. Requires cytochromes. That's not true. That's true for respiration, as we'll see. Only used by gram-positives. Not true. Gram-positives and gram-negatives perform fermentation. And then we give us 1 and 2. Number 1 is correct. This is the main drawback. Energy yield in terms of ATP per carbon is low. Okay. But 2, this is false. This is false. One thing you absolutely must remember about fermentation, no oxygen, okay, occurs in the absence of oxygen. So 2 is false. Fermentation does not require oxygen or any other external electron acceptor. The electron acceptor in the fermentation pathways is always pyruvate or something derived from pyruvate, not something else. Okay, so have to remember these basic things about fermentation. So a bit of a summary of this. So what is the problem here? Glycolysis, that is the metabolism of glucose down to pyruvate, produces too much NADH or NADPH compared to the amount of ATP. And then in order to recycle the NADH into NAD+, then you need to use pyruvate or something derived from pyruvate as an electron acceptor. Now, some of these fermentation products have got good value as for, in food production. So lactic acid fermentation is the basis of for the production of all cheeses and every kind of yogurt. Ethanolic fermentation by yeast, very big industrial, well, agricultural process used in making beer, wine, and so on. And finally, it occurs when there's no oxygen or if you've got a lot of sugar present. So when you think about it in the lab class, what you see often with the tests that we do is that the oxygen may be present for colonies growing on an agar plate, but when you have a high sugar concentration locally, bacteria will still use fermentation because the metabolic pathway is much simpler. So it's kind of easy for them. If you've got a huge amount of sugar, don't do something complicated. Just use a simple fermentation pathway and you'll be able to have enough ATP to grow quickly. So that's why if you've got a lot of sugar, fermentation will also occur in the presence of oxygen, okay? Right, so that's fermentation. Now we're going to think about respiration. Firstly, aerobic respiration, and then secondly, anaerobic respiration. And the things to, well, the basic way it works, I don't want to, have you done this in biochemistry yet or not? No, okay. That makes things a little bit difficult, but basically, okay, we've seen glycolysis, okay? Glycolysis, let's just think about glucose going to pyruvate. Forget about the metabolism of amino acids or fatty acids. Just bear in mind that these are also sources of organic carbon and they can also feed into respiration or fermentation pathways. But just thinking about glucose as the source of carbon here. Glycolysis is glucose being metabolized to pyruvate. You get a little bit of ATP, you get NADH. Then the next step in respiration is the Krebs or the tricarboxylic acid cycle. So you've got this kind of like metabolic pathway that forms a circle. It's not a biochemistry class, so we don't need to think about what is happening here precisely at all these different steps. The only thing that you need to remember right for us right now in microbiology is that what is happening is that all the carbon in the pyruvate, so this is a 3-carbon molecule, is going to be oxidized to carbon dioxide. So you throw out carbon dioxide. So if the carbon is getting oxidized, then that means it's got to give up electrons and transfer them to an electron acceptor. The electron acceptor is NAD+, so you generate a large amount of NADH, or FADH, during the Krebs tricarboxylic acid cycle. So the end result here of after glycolysis and the Krebs cycle is that you have all of the carbon in glucose is oxidized to CO2 and you get a large amount of NADH. And then the last step is electron transport and oxidative phosphorylation. So these electrons on NADH, they've got to go somewhere to regenerate the pool of NAD plus inside the bacterium. And here, the electron acceptor is going to be molecular oxygen in aerobic respiration. So during this process, the electrons are transferred from NADH along an electron transport chain. They end up on oxygen. So this allows you to recycle the NADH into NAD+, and also it will generate a proton gradient, and this proton gradient will be used to produce ATP. Okay, so these are the three steps: glycolysis, Krebs cycle, and then electron transport chain and ATP production. Okay, so in E. coli, it's quite a simple process. And there's basically three steps. So after the Krebs cycle, we're not going to go into the Krebs cycle at all. We're going to think about the electron transport chain and ATP production. So after the Krebs cycle, you get a lot of NADH. Okay, now the next step here is to take the electrons from NADH and push them onto molecular oxygen, which is a really, really great electron acceptor. It's the best, okay? So the first step is this enzyme complex called NADH dehydrogenase. So these electrons and an extra proton will be transferred through a bunch of molecules here onto quinone. So quinone is what? It's this hydrophobic molecule with a kind of lipid tail here. And quinone has got these two kind of ketone groups on the ring here. So if quinone is reduced, that is, it will take the electrons and two protons from NADH and a proton, and these ketone groups will be transformed to alcohol groups, right? So this becomes OH, and this becomes OH. Okay? So you put the electrons onto quinone. So this is a hydrophobic small molecule that is within the lipid bilayer of the plasma membrane. And this quinone, or the QH2 is the reduced form, will then take the electrons and transfer them to the cytochrome oxidase complex, which is over here. So these electrons, when the reduced form of quinone docks onto the cytochrome oxidase complex, the electrons go onto the cytochromes, but the protons, they're released into the periplasmic space. That's very important because with the quinone cycle, okay, and this regenerates the oxidized form of quinone. So the quinone just is cycling around in the membrane. Every time it transports some electrons, it will pick up protons from the cytoplasmic face of the cell and release them on the periplasmic space. So this generates a proton gradient. Okay, you have more protons here than you have in the cytoplasm. Okay, quinone cycle, and then the last step is the electrons that have come off the reduced form of quinone are transferred through different cytochromes and then finally onto molecular oxygen. So cytochromes are what? These are small proteins that are associated with a heme group here. So this is a one cytochrome. So the heme group, that's got iron on the inside, right? It's like hemoglobin. So the electron carrying part here is the iron, which is going to be, being oxidized and reduced between Fe2 plus and 3 plus. Okay, so what is happening here? So E. coli has got two cytochrome oxidase complexes, the BD complex and the BO complex. Now, if oxygen is abundant, you've got a lot of oxygen, then it will be the BO complex that is used. Why? Because the BO complex can transport a couple more protons into the periplasmic space. So it's going to increase the proton gradient. Now, but the problem with the BO branch is that the affinity for oxygen is quite low. So it's only going to function if the oxygen concentration is high. Now, for E. coli, in the gut most of the time, the oxygen concentration is very low. So it will mainly be the BD branch, okay, BO, O, oxygen. If you've got oxygen, you use BO. The BD branch has got a higher affinity for oxygen, so it works with a low oxygen concentration, but it can't really transport extra protons. So you get a little bit less of a proton gradient. However, you still get some kind of contribution to the proton gradient because every time the electrons come down through these cytochromes that are in the complex, in the end, they're transported to the cytosolic phase of this membrane protein complex. And then you get two protons react with half a molecule of oxygen, and that will give you one molecule of water. So the protons concentration, the acidity on the cytoplasmic phase is reduced. Okay, so the end result here is that through this electron transport mechanism, bacteria generate a proton motive force, a higher concentration of protons in the periplasmic phase, and this can be used to generate ATP through a proton ATPase. So these protons come from the periplasm to the cytoplasm, and this is what powers the generation of ATP. And this is very, very efficient. So if you calculate the number of protons that can be translocated per molecule of glucose, and how many protons are required to generate one molecule of ATP, then in fact, the maximum theoretical yield is 38 ATP per molecule of glucose, way higher than what you get from fermentation. So that's how aerobic respiration works. Anaerobic respiration. This is the next step. Now, it sounds like it shouldn't exist, right? Because respiration, we always think about oxygen. It's an aerobic process. And for sure, that's the only way we can generate energy. However, some bacteria can also use different types of electron acceptor for respiration. So in aerobic respiration, okay, think about it. Basically, the electrons are coming from where? They're coming from this half reaction, NADH, releasing electrons to produce NAD+. And NADH is a really, really good electron donor. Oxygen is really great electron acceptor. So that's why this process is really energetically favorable. But oxygen isn't the only good electron acceptor. So for example, nitrate to nitrite, I mean, that's pretty good electron acceptor. And nitrite going to nitrogen gas, also very, very good electron acceptor. So what occurs in a lot of bacteria is that in the absence of oxygen, they can use nitrate or nitrite as the electron acceptor. And what will happen here is, the beginning of the electron transport chain for anaerobic respiration is the same. So you've got the NADH dehydrogenase, you've got the quinone cycle, but the third part is different. The cytochrome complex is going to push the electrons not onto molecular oxygen, but onto nitrate first and then onto nitrite. And the end product in a lot of enterobacteria is nitrogen gas, N2. Okay, so that works pretty well. And this process of anaerobic respiration can use not just oxidized forms of nitrogen, nitrate or nitrite as the electron acceptor, but any inorganic, well, atom or molecule or ion, which is a good electron acceptor. So for example, using nitrate as the electron acceptor, lots of enterobacteria can do this. Same for Pseudomonas bacillus and so on. But there are a lot of bacteria that can use sulfate or thiosulfate as the electron acceptor. And this will produce reduced forms of sulfur, H2S. So everybody who was in the lab class yesterday, if you saw the black precipitate in some of the Kliegler agar tubes, this is the process that's occurring. So these bacteria are performing anaerobic respiration with thiosulfate as the electron acceptor, and the product is, well, sulfide ions, which will also react with the iron that's in the medium to produce the black precipitate. Iron sulfide, that's what you see. So oxidized forms of sulfur can be used as electron acceptors. Oxidized forms of iron can be used as electron acceptors, and also quite unusual inorganic molecules like selenate and arsenate can be used as electron acceptors by certain types of bacteria. So in anaerobic environments, like in the water table, there's no oxygen present, but there are a lot of dissolved inorganic electron acceptors. And the bacteria will tend to use the best electron acceptors first. So the best one is nitrate, so that's going to be used up first by denitrifying bacteria. Then manganese, then iron, then sulfate, and then finally the worst electron acceptor is carbon dioxide. So this is really at the bottom of the chain, and this allows bacteria, well, methanogens, to produce methane. Okay, now this is kind of a difficult concept here, and also since you haven't done really respiration and the Krebs cycle before, I think it's worth trying to spend a little bit of time understanding the process. So think about this and try and put the different steps in the right order. And if you don't know, maybe discuss with the person sitting next to you and think about it and discuss between yourselves, because it's not so easy. So don't hesitate to talk and exchange with another person, because you might need to put your heads together to figure this out. I also have to think about it myself. Okay. Yeah, I got it. Okay, we got 2 people who figured it out so far. If you don't have any idea, turn around, ask the person next to you if they've got any ideas. See if you can figure it out between the both of you. Okay, we got enough answers, I think. So let's have a look at this. Okay, so that's what people chose, and the correct answer is 3. So that's good. Good job. Now let's go through this and see why that's the correct answer. Second most popular answer is 4, which is also not bad. Also not bad, number 4. So let's think about how to figure this out, okay? So anaerobic respiration, you've got carbon, glucose as the source of energy to start off with. So the first step is always going to be glycolysis. That's the first step in fermentation. It's the first step in respiration. So first step is definitely going to be C. So it's going to be, so 2 is wrong, okay? So the first step is glycolysis. Glucose goes to pyruvate. Then what happens next? CEAD. Here we've got pyruvate, ATP hydrolysis, then protomotive force, and then electron transfer. These steps are the wrong way round, okay? Especially, well, basically all of them are the wrong way round. So D, A, and E, they're the wrong way round. Okay, so for thinking about E to A, here, if you chose 1, you're saying that first you get ATP hydrolysis. Oh, that's where you use up ATP in this case. If you've got ATP hydrolysis, then you use up ATP. But that's not what you want, right? You want to produce ATP. That's the point of respiration. That's the point of like using glucose. You want to generate ATP. You don't want to use it up. So in fact, E, A, and D, they're actually in the right order. If you use up ATP, then you can generate a proton motive force. And in that case, you could, well, then the D is wrong. But the reason this is wrong here is that E is going the wrong way round, okay? E is never going to be right because the whole point of your respiration is to generate ATP. But E says you're going to use up ATP. So answer number 1 cannot be correct because you're not producing ATP. So that leaves 3 and 4. And they both end up with A and G. So what's A and G? Generate the proton motive force and then produce ATP. Yeah, that's right. That's what you want to do. That's your end product is ATP. So both of these are correct. And then you have 2 choices. Either F, electron transfer from pyruvate to a good electron acceptor, or TCA Krebs cycle and then electron transfer from NADH. The correct answer here is 3 because step F is a little bit of a simplification of the process. So overall, that's what's happening, but in fact, there are 2 intermediate steps. Okay, first is the Krebs cycle, generates a lot of NADH, and then the electron transport chain is taking the electrons from NADH to a good electron acceptor. So that's why 3 is the right answer. Okay, so if you understand this, then I think you know enough to understand how bacteria and other organisms generate energy from respiration. Okay, so that's using carbon as an energy source, either by fermentation or respiration. Now we have to think about autotrophs, chemotrophs and phototrophs. So here we have bacteria that do not have access to organic carbon. They have to fix carbon dioxide, and so they can't use organic carbon as a source of energy or electrons. They have to use something else. So chemolithotrophs, they're using inorganic chemistry as a source of electrons and a source of energy. And there are 3 different types, basically: hydrogen oxidizers, nitrifying bacteria, and sulfur oxidizers. So all of these bacteria generate their metabolic energy and get electrons from a redox reaction. So basically, at the base of this metabolism has got to be the transfer of electrons from an electron donor to an electron acceptor. And whatever the pair of donor and acceptor, the donor has got to be further up on this scale than the acceptor. So if you remember, in anaerobic respiration, we saw that nitrate could be the electron acceptor. Okay, and that works because we've got NADH up here as the electron donor. So the electrons are getting transferred from way up here down to there. But nitrite can also be the electron donor. So then the nitrite will be oxidized to nitrate. But in order for this to work, the bacteria have to have access to a really, really good electron acceptor. So if you have oxygen available, then this can work. Okay? So somehow you've got to have a redox reaction that is energetically favorable. Transfer from an electron donor to an electron acceptor. Now, nitrifying bacteria and sulfur oxidizers, what they're doing, it's kind of the opposite of anaerobic respiration. So you have the same kind of half reactions for reduction oxidation of the nitrogen or the sulfur compounds, but the reason it works is because they've got oxygen available. Okay, there's an even better electron acceptor to make the reaction favorable. Okay, so let's have a look at hydrogen oxidizers. 2 different types, anaerobic and aerobic hydrogen oxidizers. Now, in the presence of oxygen, you have these types of bacteria. Aquifex, okay, makes water. Hydrogenophaga eats hydrogen. And alkaligenes. So if you have molecular hydrogen and molecular oxygen, this is highly energetically favorable. You know, this will explode, of course. You can burn hydrogen gas. And so this will be possible in some kind of like volcanic springs where, you know, molecular hydrogen is released from the earth to the surface. For inorganic anaerobic hydrogen oxidizers, then you have methanogens. So here you've got Methanococcus, which is an archaeobacterium. So blue, it's an archaebacterium. So here, hydrogen is the electron donor. Carbon dioxide is the electron acceptor. Remember, this is the worst possible electron acceptor. So you need a really great electron donor in order for this to work. And the only one that will function is molecular hydrogen. And this will produce methane. Now, without going into the details here, just think about the physical chemistry. This is the redox potential for the half reaction of hydrogen giving up its electrons. And this is the redox potential of NADH giving up its electrons. So in fact, molecular hydrogen is an even better electron donor than NADH. So that means that this kind of reaction will go, okay? You can directly move electrons from molecular hydrogen to NADH. So you get NADH produced directly, and then what happens? Okay, that's the first reaction in the chain. Then what happens? Well, then if you've got oxygen, then the same thing will happen as in aerobic respiration, right? So all these electrons will go down the electron transport chain, produce a proton gradient, and the proton gradient will give you ATP. Okay, so that's kind of the way it works, basically, because the molecular hydrogen is such a good electron donor. Now, how about nitrifying bacteria? So these bacteria are using relatively reduced forms of nitrogen, so ammonium or nitrite as the electron donor, and molecular oxygen as the electron acceptor. So nitrosamonas, a monas uses ammonium as the electron donor, which will oxidize the ammonium to nitrite, and Nitrobacter will oxidize the nitrite back to nitrate. Okay, so that's how you that's how I remember which one does what here. Nitrosamonas, ammonium, and Nitrobacter is the other one. Now, the problem for the bacteria here is that this half reaction, nitrite to nitrate, isn't a very good electron donor. So you can't move these electrons directly onto NADH. It's not a good enough electron donor. So how does it work? Well, what has to happen is that the only thing the bacteria can do is, if I kind of NO2 minus to NO3 minus, you've got to put the electrons, this will give you an electron here. Okay, 2 electrons even. So they've got to go to an electron transport chain, and somehow this will be coupled to proton transport. So this, and at the end, the electrons will go to oxygen. Okay. So even if there's nothing happening in the electron transport chain, you just have the final step where the electrons go onto molecular oxygen. This is going to use up protons on the cytoplasmic face of the bacterium. And that's going to generate a proton gradient. And the proton gradient, you can use it to generate ATP. Okay, just like we saw. And you can also use it to generate NADH by what's called reverse electron transport. So normally, as we saw for respiration, electron transport is from NADH to some inorganic electron acceptor. And if you've got a poor electron donor like nitrate, then you can't do it the other way around. But you can push the reduction of NAD plus to NADH to go in the wrong direction if you use energy from a good proton gradient. Why? Because, well, this is probably taking a bit too long, to be honest. But if you remember, you've got to think back to last time. You remember the Nernst equation. If you remember, if you've got a good proton gradient, this can be quantified in terms of the electrical potential across the membrane. Right? So if you've got a big proton gradient, your electrical potential will be higher than the electrical potential here that you need to push this reaction forward. Okay, so just in physical chemical terms, you can get it to work because you've produced a proton gradient, and this gives you an electrical potential which allows you to push this redox reaction in the wrong way. Okay? Right, so that's nitrogen oxidizers. Sulfur oxidizers, how do they work? Well, they're using reduced forms of sulfur to as a source of energy. The electron acceptors are either molecular oxygen or nitrate, because in fact nitrate is a better electron acceptor than sulfate or sulfite. So there's two interesting things that can happen here. So one of them is that sulfite can be oxidized directly by molecular oxygen. This is highly favorable, so you can produce proton gradient. You can produce a proton gradient, and then by reversing it, and then you can get ATP by this process, and NADH by reverse electron transport. The other thing that's kind of interesting, especially for anaerobes, is that you can produce ATP directly by a kind of series of reactions that switches around sulfate for phosphate on an adenosine monophosphate molecule. So you can get ATP produced directly by this oxidative sulfur metabolism. Okay, now, once again, all of this is kind of complicated, and we've done it a little bit quickly. One of the things that I find is important to understand is which couples can work and which couples can't work, okay? So think about this, and what you'll need to answer this question, perhaps, is the kind of scale where you've got the redox potentials of the different couples. Okay, pretty good. Let's have a look, see what people say. So a lot of people chose one. Correct. Yeah, this works. This is anaerobic respiration. It works. Glucose, good electron donor. Nitrate, good electron acceptor. That works. Similarly here, question 5. Molecular hydrogen, extremely good electron donor. Nitrate as an electron acceptor. Yeah, that works. That will be energetically favorable. 2, nobody chose this. Good. That will not work. Okay. It can't. It's not possible. And that leaves us 3 and 4. 3 actually does exist. It's called sulfur disproportionation. It's a bit strange, but in fact it does exist. But you know, of course, very difficult to guess that. So I don't think you could really have got this right. Number 4, This is a wrong answer. Okay, why is it the wrong answer? It's because nitrate is a better electron acceptor than sulfate. So that means that ammonium is a pretty poor electron donor. Nitrogen oxidizers, chemolithoautotrophs, can only live if they have molecular oxygen, a better electron acceptor than oxidized forms of nitrogen. So sulfur oxidizers can use nitrate as an electron acceptor, but ammonium oxidizers cannot use sulfate. Okay, it only works one way round because it's only energetically favorable in one direction, and that's important. Okay. Okay, right. So I think not obvious, but an important conclusion to draw. Okay, so we have 20 minutes. So what I think I'm going to do is probably just try and talk about carbon and nitrogen cycles. And then rather unfortunately, pretty much like every year, I'm going to miss the photosynthetic bacterial part. And I've already recorded it from previous years, so that will just have to be like check out the recording, because I'm afraid pretty much every year I never get to the end of this lecture. So thinking about the carbon cycle and nitrogen cycle. Okay, so probably everybody knows this. You've got a cycle from carbon dioxide is incorporated into organic carbon by carbon fixation, either by plants, by oxygenic photosynthesis, or by autotrophs. Like these chemolithoautotrophs also incorporate CO2 into organic carbon. But in quantitative terms, you have about 10,000 times more carbon or CO2 absorbed by photosynthesis than by chemolithoautotrophs. And in terms of the mix between plants and microbes, that is cyanobacteria basically, then it's about 50-50. So cyanobacteria in the oceans, they'll be fixing about half the CO2 that is taken up in any given year. So all of this will give us organic carbon, which then is going to be recycled back to carbon dioxide, either through respiration or through anaerobic respiration and fermentation in anaerobes. Now, some of these anaerobic fermentation pathways will produce molecular hydrogen, and as we saw, methanogens will combine this molecular hydrogen with CO2 to produce methane. And then this methane can be oxidized to CO2 by methanotrophs, other types of bacteria that use methane as a source of carbon and energy in the presence of molecular oxygen. So what I'm going to talk about next is what types of organic sources of carbon can be used as an energy source by bacteria, because up till now we've just been talking about glucose. So I want to talk a little bit about other sources of carbon that are not so easy to digest, like cellulose, lignin, and hydrocarbons. And then I want to talk a little bit about methane generation and methane oxidation, because as you know, methane is itself an important greenhouse gas. Okay, so for sources of carbon that can be metabolized, so pretty much everybody or every organism can metabolize starch, amidon, which is a polymer of glucose in these linkages that can be easily digested. However, cellulose, it's a different question. So basically no animal has got enzymes that can break down this beta 1 to 4 linkage between the glucose molecules in cellulose. So herbivores rely on their gut bacteria to do this for them. Okay, so in ruminants, there are gram-positive gut bacteria called, for example, ruminococcus. Let me just check that I've got this right here. Yeah, ruminococcus in the rumen of the cow's guts, which will be fermenting cellulose by breaking down this beta 1 to 4 linkage. Okay, so how do the cows then, or other herbivores, get the energy from this? If the bacteria are breaking down the cellulose and fermenting it, how do the cows get some nutritional value? Well, the gut bacteria, like ruminococcus, will produce fermentation products like acetate, lactic acid, and these are absorbed into the bloodstream of the cow. And then because we've got like our circulation and our cardiovascular system, we've got molecular oxygen everywhere around our bodies because of hemoglobin, right? So then this lactic acid, acetic acid can be absorbed and then fully oxidized by the Krebs cycle to allow us to generate energy, or us, I mean like cows, other animals. So it's, you know, commensal bacteria in the guts of herbivores that allow animals to digest cellulose. And that's because both anaerobic and aerobic pathways exist for the digestion of these carbon compounds. Now, lignin, which is the major polymer in wood, is much more difficult to digest, okay, because the only pathway that allows this polymer to be broken down relies on molecular oxygen. So it's only aerobes that can break this down, and not bacteria, in fact. It's filamentous fungi which can break down lignin. So if you're walking around in a forest and you see a log that's fallen down, you'll see a load of fungi growing on the surface, and they live by breaking down the lignin. Now, otherwise, there are no anaerobic pathways to break down wood, okay? So that's why, you know, if trees fall down in an anaerobic environment and the wood gets buried, then the carbon will be sequestered, basically forever. So that's where all the coal deposits in the world came from, because there's no anaerobic pathway that can break down the lignin. So that's why it just hangs around and is sequestered. Now, the last thing that's kind of interesting about carbon sources for bacteria and other microbes are hydrocarbons, like petroleum products, things that are derived from the chemical industry. Now, these are not naturally occurring polymers, but there are still some bacteria that can break them down. So a lot of soil bacteria, like maybe Pseudomonas, can break down synthetic hydrocarbons. God damn it, why does it keep doing this? Now, so most of these degradation pathways require molecular oxygen, but there are some which can work in an anaerobic environment. So this is a kind of field of bioremediation for polluted soils, this kind of thing. You can use bacteria to degrade hydrocarbons if you have a good electron acceptor present, like nitrate or sulfate. Okay, now just want to think a little bit about methane production and use, because methane is really a very, very significant greenhouse gas, and bacteria are responsible for a lot of its production. So the basic metabolic pathway used by methanogens is to use molecular hydrogen as the source of electrons and energy, and carbon dioxide is the electron acceptor. And this produces methane. And this pathway is energetically favorable because molecular hydrogen is a better electron donor than methane. Okay, so this works. The redox potential is favorable. So most, I think all methanogens are archaebacteria, are archaea, and you get methane production where you have no oxygen, but you've got abundant organic carbon. So the organic carbon will be fermented by other bacteria, and molecular hydrogen will be one of the fermentation products, as well as CO2. And then methanogens will combine these to produce methane. So this occurs a lot in ruminant guts. And that's why, animal farming is a significant producer of methane and greenhouse gases. It also occurs in marshes and in coastal ocean sediments. So in coastal sediments, especially in the Arctic, a lot of this methane is immobilized in kind of complexes with frozen ice, and which are called methane clathrates. And this is a real worry, because if the temperature of these coastal sediments increases, then what's going to happen is all of these methane clathrates are going to release their methane into the atmosphere in a very, very short period of time. And this will give us a kind of positive feedback mechanism for greenhouse warming. And this is a real concern here, because methane is 25 to 30 times more potent than carbon dioxide, not that. So this is one of the feedback mechanisms that we could shortly be confronted with. Now, it's not all bad news from bacteria. So bacteria, methanogens, are the source of methane. But there are other bacteria that consume methane. Okay, these are methanotrophs. Now, methanotrophs are also archaeobacteria, and they often don't grow on their own. They need to grow with other types of bacteria in a symbiosis. So, for example, you'll have the methanotroph, which is oxidizing methane to carbon dioxide. This will give it a lot of electrons, but it's got to find an electron acceptor in order to keep cycling the metabolism. And that's the role of the other bacterium in the symbiosis. So, for example, methanotrophs can live with sulfur-reducing bacteria, and the sulfur-reducing bacteria have got the ability to transfer the electrons produced by the methanotroph onto sulfate. Now, the question is, how does this electron transfer occur between the two types of bacteria? Now, what is interesting in this particular consortium, this particular symbiosis, is that the sulfur-reducing bacteria, which are gram-negative proteobacteria, which are called H here, A is the archaeobacteria. So these sulfur-reducing bacteria produce type 4 pili that make contact with the archaeobacteria, and these are like wires that are conducting the electrons from one bacterium to another. So they actually push out the pili, and then the electrons flow backwards. So they kind of make a little kind of electrical circuit between the two types of bacteria. So, maybe all the methane from the methane clathrates will not actually be released, because methanotrophs will grow, because they'll suddenly find they have a good source of methane to grow on. So it will still release carbon dioxide, but perhaps the greenhouse effect will be a little bit less, because it's a less potent greenhouse gas. Okay, so I've got only 5 minutes to talk about this. I might actually record this as well, and I'll give you, what do you think? Should I try and rush through this, or should I just record it? Okay, so I'll try and record this, because I think it will take a little bit of time to go through these. No, let's just do the first part, okay? There's a bit here and a bit of a question, okay? So we have enough time just to do the first 2 slides. Then I'll record the rest. Okay, so the nitrogen cycle. Also, bacteria are involved in all steps of the nitrogen cycle. So the nitrogen cycle is what? You have oxidized and reduced forms of inorganic nitrogen, as well as nitrogen gas, and they're being cycled between these different forms. And also, nitrogen can exist as an organic form, okay, in amines, amino acids, and nucleotide bases. So all living organisms, all bacteria, need to get organic nitrogen somehow. So a lot of bacteria will reduce nitrate first to ammonium and then incorporate that ammonium into their amino acids. Some bacteria, nitrogen fixing bacteria and cyanobacteria, can reduce nitrogen gas in the atmosphere into ammonium and then incorporate that into amino acids, like azotobacter and cyanobacteria. Now, these other forms of so this is for using nitrogen in biomolecules. So these other processes are transforming inorganic nitrogen from oxidized to reduced forms as part of the energy metabolism of the bacterium. And that's really what the arrows are about, okay? So the different colors of the arrows represent different things in this diagram. So what is the process that's occurring with the white arrows here? Yeah, just think about that. Let me set up the question. And once again, it's not obvious. You might need to talk with the person sitting next to you and exchange ideas and see if you can figure it out between you. Because, you know, one of the confusing things for me, okay, I have to say this personally, is you've got the arrows going in both directions. So you think, how can this work, right? How can bacteria get energy with both processes? Somehow doesn't make sense. But it does make sense. You have to just really understand it. Okay, we got 2 minutes, so I better put the answers up. Okay. This is what you've put in. These are the answers. So this is what people have sent in. Answer 3 is the correct answer. So well done. You figured it out. What is happening here is that reduced forms of nitrogen are being used as a source of energy and electrons. So this is Nitrosomonas, Nitrobacter, and it only works in the presence of molecular oxygen. So fermentation, no. Anaerobic respiration, no. Aerobic respiration, no. Aerobic respiration, that's with oxygen, right? Doesn't involve nitrogen at all. So aerobic respiration, that's wrong. Fermentation is wrong. Okay, that's good. Anaerobic respiration, that's wrong, but that's the purple arrows coming down here. That's anaerobic respiration using nitrate and nitrite as the electron acceptor. And the reason this is energetically favorable is because the energy isn't coming from the redox reaction. For nitrate being reduced to nitrogen gas or nitrite and ammonium, the energy comes from the carbon source. Okay, so that's why you can have the arrows working in both directions. For the purple arrows, the energy source is the carbon, but for the white arrows, it's this redox reaction, which is energetically favorable because molecular oxygen is present. Okay, so I'll just stop there. Microbe of the week tonight, right? Yeah, tonight or Sunday night? What do you prefer? Sunday night would be better, right? Because Friday you'd probably be doing other stuff. No? Yeah? Yeah. Sunday night, 7 p.m., I'll send out a microbe of the week. Okay? Goodbye, everybody, and I'll put the recordings up for the rest of the today's lecture. Anybody wants information about Birmingham, email me. I'll try and answer. Birmingham.